Well, let's move on. I call the next speaker to here. He's Director of Research of the ACE Foundation and an expert in the genetic basis of AD. Thanks, Agustin, for being here. And he's going to talk about dementia GWAS in the Spanish population.
Okay, thank you very much to Cibernet, and thank you very much especially to the organizers for inviting me today to present the results of the GESCO consortium, and on behalf of them and also the Fundación ACE Genome Research Project. And double thanks because today is the Alzheimer's Day, and it's a very important day for the Foundation. As you may know, we are devoted to the management of this problem in our environment. And the only thing I can say is that, we have 1 million people with neurodegenerative disorders in Spain. That in turn means that you have more million people afflicted with the problem, caregivers and families, and the only thing I can do is to offer this conference to them. And let us start speaking about the GESCO. The GESCO is a down-to-top organization. Was a dream held by Pascual Sanchez Juan and a small group of researchers of Spain, 11 groups initially, that wanted to work together. And now we are organizing our production. We have several papers out, and we are probably much more to come. And also the community is growing a lot. In fact, now we are 20 groups working around Spain, and they are involved in 9 communities. Hopefully in the future, we are very open to receive more groups around Spain and even outside Spain as well. So we are very happy that the consortium is growing and working relatively well, I would say. I'm going to start talking about a big project we have in our hands and is running at this moment is the GRACE, the GESCO GWAS project. I will provide you an overview of the project. The project is still running. And I will specifically focus in the stage 1. It's based on specifically on the cases of dementia we have in the Fundación ACE. This is the stage 1 of the project. And after that, I was explaining a little bit how we are progressing in the GESCO stage of the project. And at the end, I provide conclusion and future directions. Okay, the GRACE, the GESCO project, what is it? But actually, it's a 3 years multi-center, 2-stage research project focused on the etiology. Etiology means that we are looking for causality networks related to the disease. That is it's important to reinforce it. And also looking for not only Alzheimer's dementia, but also related disorders. And probably, as mentioned yesterday by Ángel Carracero, is the largest GWAS effort in Spain of any disease at this moment. And we wanted to increase and grow also the data we are generating to have a more clear idea in the future about these causality networks. And this is the original plan. And the original plan obviously was based on the money, of course, because we have money initially to run a GWAS of about 12,000 samples, and we decide to focus the initial stage in the decoding on a single site cohort or data set coming from the foundation, because we have, as you may know, a very large biobank available for this. And after that, we will try to replicate the results in stage 2, pulling together the information and the biobank coming from the rest of the groups from Spain. And actually, we have the samples available. And in fact, based on the increase of the money we have, of course, and also the increase of the sample we are receiving, we can announce that the pH will be bigger, probably 50% bigger in the developing or decoding of the samples. As you can see, at the end of the stage 1, we have 7,400 samples decoded in the stage 1, and we are planning to run a GWAS of more than 10,000 samples in the second stage. And in fact, we have half of the samples in the second stage already available and decoded at this moment and in this month. So probably we are going to to finish the decode of the samples earlier than we expected. And this is a very hard work done in our laboratory for QC of the DNAs, and also a very hard work in the National Center of Genotyping, Angel Carracedo team working together with us on this stage. And why this is a different project? This is also interesting to reinforce. As you can see here, the sample size of cases. Whoa. I think that I know what is the problem now. This mounted. Okay, you can see here that we have 4,100 cases coming from a single site. What means that we have a single site for evaluation? This is a very valuable situation compared to the rest of consortium around the world, because they are pooling samples, diagnosis by different methods, by different professionals around the world. In our case, we have the same diagnosis criteria for all the samples available. This is a very, very important issue to be taken into account for the rest of the speech. Of course, we hope, and I think that we have an expert clinical phenotyping, and we can check for the comorbidities on the patients. We can check for other cognitive problems, and we can identify. This kind of information is never in the data sets of the Genome-wide Association studies previously published. And of course, we have also a follow-up information of many of our patients as well, and that means that we can also analyze genes related to disease progression. I think is a very interesting characteristic of this project. And of course, we can, in the future, also link the information of the drug taken by the patients, and also conduct pharmacogenetic studies. And this is also important from the genetic point of view, because we know previously that we have a very homogeneous population. In fact, we have several GWAS previously published in Spain. We ran years ago. And we know that we have a very consistent structure of the population from a genetic point of view. And as you can see here, it's not working. Okay. Yeah, as you can see here in the, as you can see here, we have a subcontinental population. What means that? That 99% of the samples we run in the stage 1 are located in the South European ancestry. So compared to the GWAS run by Americans, for example, they have a much more stratified population, and they have a lot of noise related to the structure of the population. Our population is much more homogeneous. Maybe we have macro stratification that may affect to the identification of rare variants, but actually now we have technology developed by Angel Carracedo even to tackle with the macro stratification of the Spanish population, because now we know the bars and the different ancestries we have inside the peninsula as well. So this is the range of subcontinental structure is very valuable for to generate good signals related to the causality of the disease. And now let's talk a little bit more on the phenotypes. Sometimes when you reach data related to GWAS, what you have is a 0-1 for the phenotyping. You have the dementia or the Alzheimer's disease, or you have not. But actually, this is not true. We have not a single disease. The people have more than one disease running and provoking damages in the brain. So what means is that we can characterize and divide the population based on the comorbidities and other findings that they may have in the brain. And then in the more simplistic way, we organize the information based on the vascular burden of our patients, because we have already the information, and we can conduct endophenotype analysis that never have been done at the GWAS, the Genome-wide Association level, never. So we can add the level of certainty of the vascular burden and the level of certainty of the neurodegenerative process to the to stratify the population in different ways that we did in previous years. Okay, let's talk about a little more about the methodologies that, we have to manage big numbers of samples, and we have to be very restrictive for the QC, the quality controls in the laboratory to have a successful genotyping. And we did, we performed a parallel analysis based on fluorescence to quantify and also to look for the integrity in lab chips to check that the DNA is properly and have a good quality for running the GWAS. And the genotyping, we have on board the National Center of Genotyping. They have a strong expertise to manage these kind of projects. And also they have, I have to say, they have developed a very, very interesting technology. This is the Affymetrix array that has also the variability specifically related to the Spanish population. And we think that this is very valuable to have also integrated this information in the array we are running. And after that, of course, we have a very strong bioinformatics pipelines, to clean the data and to prepare everything to run the genome-wide association studies. You can see here the parameters. I don't want to go into the meat of the matter, of course. If anyone wanted to get inside about the pipelines, please put an email to me and we can, offer any information you request. And now, of course, during this process, we have to prune, because as you can see, we genotyped 8,400 individuals, and at the end of the project, we have only 7,400 available for the GWAS. This is because you are detecting some technical problems in the GWAS generating a specific samples you have to remove from the GWAS, and also you have, of course, have to do resampling, demonstrating that you can track your samples around the experiment as well. So you have to repeat some samples and check that you have identical results at genetic level on that resampled sample. And of course, we found out an interesting thing, that the sons of the people with dementia are coming to our open house initiatives because they have the awareness of the disease. And we found out several relatives coming like controls because they have no any symptom, and we take the DNA, but we can never use them during the GWAS analysis because actually they are related to the cases. But this is bad news, you are reducing your power, but very good news for the future because we can conduct real genetics project looking for transmissions, not only of alleles, but also polygenic risk scores in these populations. And also you have to clean as well the arrays, the markers you want to analyze, and you can see we are starting with about 814,000 markers in the beginning, and at the end of the pruning of the markers based on the quality control process, you are taking only 600,000 markers for imputation. This is because we only use a common variation for the conventional GWAS analysis. We are removing all the rare variants and also very specific markers put in the array for quality controls. And as soon as you finish what is the quality control of the samples and markers, you have to start with the imputation, and this imputation is not a bad word in genetics, nothing to do with the politics, of course. And what we are doing is making the inference of the entire genome based on the data set of markers we genotyped. What means that we can make inferences of genotypes around the genotyped SNPs in our population. And we are using the HRC reference panels. It's a very huge human resource, and the Michigan server to do this kind of imputation. And this in turn converted the original 600,000 markers in 7 million SNPs available for evaluation in our population. So you have 7,400 people with 7 million SNPs ready to go for analysis. And after that, you can do the association study using conventional technology. Don't want to enter into the details so well. And at the end of the project, you can meta-analyze your results with previously available data sets as well, using the meta-analysis approach. So the first thing is to select a good model, a statistical model for your project. And this is the statistical model is based on the noise you are finding out in your population. If you never covariate these GWAS, you have an inflation factor about 14%, which is not bad, but this is due probably to the macro stratification and the, because we have also a fraction, a small one, but a fraction of Latin Americans living in Spain that they are also in our data set. And we have to remove, or alternatively, you can control this noise and reducing to 3%, merely using 4 principal components of SNPs to control this macro stratification. And you can see in our case, modeling with age and gender is not a good idea. You have deflated inflation. That means that you have less positive than expected by chance. That means that this model are not good, at least in our hands, and we decided to use this one for the rest of the project. And this is the original Manhattan plot. This is ApoE, of course, and this is the control, the positive control of the signal. If you never seen this kind of signal in your Manhattan plot, you have to throw away these GWAS, because in all case controls for dementia have to appear at the strongest signals of ApoE. And you can see here you have 10 to the minus 63 p-value for this association, but also confirming other genes related to the disease. We have a nominal association of them. That is a good news, indicating that the data set, the configuration, is signaling or pointing to Alzheimer's disease genes. But, let's use the classification of patients. In fact, this never has been done earlier because the information is not available. And we can imagine that you may have genes previously known, the 20 genes mentioned yesterday by Angel. We are using more, that maybe have a behavior like we said, adilocyte, where you can see that the effect size, the risk of the genes is increasing in the more pure forms of Alzheimer's disease. So the people who has not a vascular burden, we call that kind of group adilocyte. And also maybe there are another kind of genes that irrespective of the vascular burden, you have the same effect size on every stratum of the endophenotypes. And maybe you have another group of genes, this is a hypothesis, that maybe are increasing the risk when you have the people with more vascular burden. This is a very, very ancient, not relatively, 9 years old hypothesis indicating that maybe you have 2 axes of causability of the disease. And we try to reproduce this. And actually what happens is that actually that kind of group of genes exists, and we detect it. We have AD-like loci. You have a small list here. You have neutral loci with a flat risk in the different endophenotypes, and also you have genes specifically related to the vascular burden. And this never has been done. We established analyzing co-regulatory networks of these 3 subsets of genes. We found out these pathways related to every gene. And this was our first surprise. The AD-like genes, we hope to find out amyloid, we have to find out the brain functions, etc. But what we found out was the vasculature development. And what we found out was the blood vessel morphogenesis as an enrichment of the genes more related to the disease. And this has to be to take in mind. And the others are related to inflammation. And the relation with vasculature, we have checked other data sets, and we have demonstrated relation of Alzheimer's disease causality networks with the heart structure phenotypes. For example, this is a paper now under review. So, this is to contrast the information of cardiovascular risk with Alzheimer's disease. We use other data sets and demonstrate that, in fact, we have a network in common between the heart structures and the Alzheimer's disease. And now a little bit about the Brainstorm Consortium findings. You can see here that almost all psychiatric disorders are related among them, and they have a common network of genes, but in the case of neurological disorder, you have not relation about causality networks among them. That means that, most of the psychiatric disorders, this is the Brain Consortium work, are related to network of genes related to the function of the CNS, and most of the neurological disorders not. They have an external causality network unrelated to brain functioning. This is based on an experiment in 1 million people, GWAS analysis in science, members of CyberNet, and our group is part of this project. And of course, we did meta-analysis, has been commented already, nothing to say. This is a cardiovascular risk gene, in fact, is the statin target. We detected and we validate the results of Maria Jose Bullido, of course. And now we are running a series of meta-analysis, and now we have detected more than 6 independent signals. Some of them are going to be published in the consortium IGAP, and we will be part of this project, and we have also a very good position in this huge paper discovering new genes related to the disease, 5 novel genes to be published very soon. So in conclusions, using our GWAS, we find out different groups of AD loci and pointing to external causality networks. External means cardiovascular inflammatory networks that may be working in the brain, but actually are systemic in the system. We confirmed different loci related to Alzheimer's disease. We find out several new genes related to the disease, and we are still exploring the IGAP data set to look for more genes. And this is only to mention that the second stage is ongoing. We're planning to run more than 10,000 samples to validate our results. And we are working also with international data sets coming to the HESCO project. That is a very good news. We are receiving samples from Tunisia, samples from Chile, and we plan to decode those samples as well. And thank you very much for your attention, of course, to our sponsors and the National Center of Genotyping and the National Biobank of Spain. Thank you very much.
Thank you, Agustin. Questions? No. I have a comment and a curiosity. In thinking about all these studies which have a very high value, and considering the plasticity of the brain, would it be possible to somehow associate these studies with some sort of measurement of the potential resilience of the brain, maybe with academic records like the IRIS cohorts? Is anyone looking into that?
Yeah, this is a great question, because actually when you are seeing the causality networks, you are thinking about where is the brain here? The configuration of this kind of data sets is to look for risk factors, and probably the resilience network and the resistance network have to be explored in a different way. And in fact, in the Brainstorm Consortium, we have demonstrated also that you have a strong correlation, genetic correlation between intelligence and Alzheimer's disease. So what we think is that, you have a systemic exposome of risk factor acting in the brain, and you have also I would say, resilience and resistant network of genes trying to buffer this kind of damage coming from in a systemic way. But one point, we need to look for endophenotypes beyond intelligence to look for this kind of resilience networks.
Maybe even long.
We need the phenotype.
And, oh, gosh, I forgot a word. This is an example of, okay, questions?
Having in mind that environmental factors can modify so much your genetic expressions, how stable are your samples?
Stable in which way?
In terms that you take the sample of one person, let's say this person is a 20 years old man, right?
Yeah.
And this 20 years old man can change their genetic expression when he's at his 40s, for example, or his 60s.
Yeah.
So do you keep this in mind in terms of...
Yeah, of course. In fact, we are looking for the way to look for gene-environmental interaction. The DNA and the genome, of course, is mostly stable, of course. And this is not a problem for genetic studies, actually looking for, the inheritance behind a trait. But I agree with you. We have to look for the way to integrate the gene-environmental interactions, to look for this kind of, combinations of genes and factors coming from outside to increase the risk of the disease, or even to protect you. But at this moment, I have to say that the mathematical modeling we are using are not working too much on this way, and we have to improve, the way to analyze this kind of interactions, because it's very complex from a mathematical point of view.
You have mentioned that you have found out 3 different types of etiological Alzheimer's disease, different types of Alzheimer's disease from a genetic point of view. My question is, well, I have 2 questions. How robust is this conclusion? How similar, or how could it be compared with other proposals of subclassification of Alzheimer's disease? And the third question is, how could it be validated, for instance, from the point of view of the biomarkers, from the point of view of other clinical or pathological approaches?
Well, and the first point is, the cardiovascular relationships are beyond the findings coming from genetics. Well, now we are detecting using correlation of networks and comparing the irritability and the set of genes related of each disease, what is the commonalities between the diseases. The genetic correlation with the cardiovascular networks is emerging now, very clear. In fact, in the incoming paper that is submitted at this moment in Nature Genetics by the IGAP, we have found out as well the relation with the cardiovascular and genetic correlation. So we have an independent replication of this. And also related to co-regulatory networks, we can use, we will try to replicate in a new data set coming from the GESCO, of course. And at this moment, what I have to say, this is for cardiovascular connection, we have 10 to the minus 7, which is a not bad p-value. And for inflammatory networks in the flat model, the genes with the behavior of the flat, we have a p-value of 0. That means that network, I think that we don't need more. We cannot calculate the p-value in this association. So the inflammatory path at this moment, not only based in our results, but based in many other papers, especially the paper coming from IGAP, the GESCO is part of it, published last year in Nature Genetics. I think that the inflammatory path is very clear, associated as a causal network of the disease. Causality and causal network never invalidate pathophysiology. So what is happening in the brain after, the damages coming from causality networks, it has to be studied in other ways, and you are doing. We have a lot of work ongoing on that. We are strictly speaking on etiology.